Hello friends, I am Gaurang Makwana. I welcome you all to my another video tutorial Biotech guide. In previous video lecture, we have discussed on design of a bioreactor or fermenter. In today's video lecture, we'll discuss about types of a bioreactor or fermenter. Also, we're going to discuss about the difference between bioreactor or fermenter. So today's video going on very interesting level. Stay tuned with us and enjoying this video lecture. Let us discuss first difference between bioreactor or fermenter. Mainly, bioreactor or fermenter differentiated on the basis of purpose, design, application, and the flexibility. First, purpose. A fermenter is specifically designed for a fermentation processes where microorganisms such as bacteria, yeast, or fungi are grown and used to produce various substances like ethanol, antibiotics, enzymes, or organic acids. Whereas a bioreactor is a more general term that encompasses various types of vessels used for a wide range of biological processes. Not limited to fermentation, bioreactor can be used for fermentation as well as for other processes like a cell culture, tissue engineering, or a bioprocessing. So major difference is fermenter is designed for specifically fermentation processes, or a bioreactor is for a biological processes. Then second, design. Fermenters typically have specific features tailored for microbial fermentation, such as agitator for mixing, aeration system for supplying oxygen, temperature control system, and pH monitoring and control. Whereas bioreactor can vary widely in design depending on the specific application. They may have different configuration, sizes, and components tailored to the requirements of the particular biological process being carried out. So second, design. On the basis of design, mainly fermenters have a specific features which required for a normal fermentation process like agitators, then aeration, and the pH controlling system, and all the system which required for fermentation process is specifically attached to the fermenter. Whereas bioreactor, they mainly have a vary depending on the specific application which we have discussed in types of a bioreactor or fermenter. Then application. Fermenters are primarily used in industry like pharmaceuticals, biotechnology, food and beverages, and biofuels for the large scale production of a microbial products through fermentation. Whereas bioreactors have a broader range of application beyond fermentation. They are used not only in industry mentioned about, but also in the areas like wastewater treatment, environmental bioremediation, and research laboratories for various biological processes. That means mainly fermenter having application, majorly having application in industry, whereas bioreactor not only industry, they having a broader range of application like we have discussed here, wastewater treatment, environmental bioremediation, and research laboratories. Then fourth, flexibility. Fermenters are often designed with a specific features optimized for a microbial fermentation and may not be as adaptable to different types of biological processes. Whereas bioreactors are designed to be more versatile and adaptable to different types of biological processes, allowing for a wider range of application. That means fermenters are having specific features for a specific microbial fermentation processes, whereas bioreactor are having more versatile and having wider range of application. Now we are going to discuss about types of a bioreactor. As we discuss in difference between bioreactor or fermenter, any biological process is required a specific or a versatile adaptable bioreactor. But in the case of any fermentation process, normal fermenter which have mixing components like impeller for agitation, then sparger for aeration, then the various controlling probe, then the inlet and outlet unit. Those all are important controlling unit or devices which is required for any kind of fermentation process are attached. But in the case of any kind of bioprocesses, mainly a specific design or a versatile adaptable bioreactor is required. So Now we're going to discuss about the types of a bioreactor. So majorly 7 types are there. First one is a airlift bioreactor, then continuous stirred tank bioreactor, then a photobioreactor, bubble column bioreactor, fluidized bed bioreactor, packed bed bioreactor, and a flocculated cell reactor. So majorly I covered brief design and then majorly focusing on advantages and disadvantages of this bioreactor. So let us discuss. We'll discuss one by one. First, airlift bioreactor. So the entire reactor is divided into two halves by a draft tube. Here you can see this is the draft tube, and the two halves is first inner gas region is riser, this one, and the outer ungas region downcomer, this one. Basically, what happened here? Density gradient between riser and downcomer, and the circulation is happen. Here you can see in diagram parts of LR, riser, downcomer, and then base which is connected to a nozzle bank or a sparger to pump the pressurized air. Then the headspace, this one is known as also known as a gas release region, and the gas separator is mainly used for the separating gas. Then now on the basis of this draft tube, basically two types of ALR is resulted. This one internal loop ALR and the external loop ALR. So in internal loop ALR, circulation takes place in the straight line, and in the case of external loop ALR, circulation takes place in a separate and a distinct zone because of draft tube. Draft tube is attached in middle position in the internal loop ALR, and in case of external loop ALR, draft tube is attached at the outer side, not in the middle side. So on the basis of this draft tube, two types of ALR is resulted. Then advantages and disadvantages: less maintenance, less risk defects, then easier sterilization, low energy requirement due to gradient circulation happening, then no moving part is there. Then disadvantages: greater air throughput and a higher pressure is needed, inefficient break the form, and no bubble breaker is there. Second, continuous chilled tank bioreactor, which is similar like to an ideal fermenter, having a cylindrical vessel with a motor, then central shaft is there, then supports agitator, impeller is there. Then the number of impeller is variable and it depends upon the size of the bioreactor. Then air is pressurized through a device is known as a sparger. Sparger is also there. Then is mainly depends on the variable on the basis of a types of bioreactor, like single orifice and the other types of sparger is there. Then along with a sparger, impeller enable to better gas distribution. So here you can see in the diagram ideal fermenter, which is also considering as a continuous chilled tank bioreactor. Then advantages: flexible and a low operating cost and investment need, simplicity of a construction, easy to clean and sterilization, then a good control over parameter and environment, then efficient gas transfer to growing or mixing the contents. Then the disadvantages: the need of a shaft seals and bearing, then the forming is often problem, and the consumption of a power is more due to the mechanical pressure pumps. So these are the advantages and disadvantages of a continuous chilled tank bioreactor. Third, photobioreactor. The word photo describes the bioreactor properly to cultivate phototropic microorganism or organism which grow by utilizing light energy. Phototropic organism use the process of photosynthesis to build their own biomass from the light and carbon dioxide. Phototropic organism like as a plants, mosses, then the microalgae, cyanobacteria, and the purple bacteria, which is grown inside this photobioreactor. Then photobioreactor PBR allow much higher growth rates and the purity levels. Basically, two types are there: open system PBR and the closed system PBR. Then advantages and disadvantages: higher productivity, more uniform temperature, better control of a gas transfer, reduction of evaporation of a growth medium are the advantages. And the disadvantages: cost is very high, the technically difficulty in sterilization, then enclosed photobioreactor productivity is not much more preferable. Then fourth, bubble column bioreactor. Bubble column are devices in which gas in the form of bubbles comes in contact with a liquid. The purpose may be simply to mix the liquid phase. Also internal devices such as the buffers impeller may be placed in the vessel to improve the mass transfer. Basically, gas is passed at the base of the column through perforated pipes, plates, sintered glass, or a metal microporous parger. So this is the simpler design of a bubble column bioreactor. Here you can see gas parger is there, then also gas phase is there, then the product gas and the feed gas. Then higher volumetric productivity, self-regulating, better utilization of area, and the flow distribution are the advantages. Then less efficient than other bioreactor, does not have a draft tube, then a higher installation cost and difficult to design, and a higher catalyst consumption are the disadvantages. Next, fifth, fluidized bed bioreactor is comparable to bubble column bioreactor except the top position. The top position is expanded to reduce the velocity of the fluid. Basically, in the design of a FBR is such that solids are retained in the reactor while the liquid flows out, and the bioreactor are suitable for carry out the reaction involving fluid suspended biocatalysts such as enzyme, immobilized cell, or a microbial flux. Here you can see in design or diagram, upper top position or top base is known as a disengagement zone, and also catalyst feed is there, then gas feed is there, and then non-reacted gas is flows out of the disengagement zone. Uniform particle mixing, uniform temperature gradients, and the ability to operate a reactor in a continuous state are the advantages, where pumping requirements and the pressure drop, then particle entrainment, lack of a current understating, then pressure loss scenarios are the disadvantages of this fluidized bed bioreactor. Sixth, packed bed bioreactor. Reactors which are packed with immobilized enzymes or microbial cells as a biocatalyst. Basically, these biocatalysts are present over here, solid particles on the surface of a solid particles. This solid may be used porous or a non-porous, or also may be in a rigid in nature, and the nutrient growth flows continuously over the immobilized biocatalyst, and the resulted a product is obtained in the packed bed reactor are released into the fluid and the removed. Or basically, while the flow of this fluid can be upward or downward, basically downflow under gravity is preferred, and the concentration of the nutrient can be increased by flow rate of a nutrient medium, and the pH of this medium may be maintained by the addition of a acid or alkali. So this is the simplest design of a packed bed bioreactor. Here you can see an design of a packed bed bioreactor. Biocatalyst is present over here there on the solid surfaces, is known as a bed, and the mainly nutrient growth is come in this direction, and the mainly react to the biocatalyst, and the product is result, and this product is mainly retained at the bottom downward direction or at the bottom. Now the advantages and disadvantages. So low operating cost, continuous operation, catalyst stay in a bioreactor, simple design, and effective at the high temperature. Those all are the advantages of this packed bed bioreactor, where the undesired heat gradients, poor temperature control, difficult to clean, and difficult to replace the catalyst are the disadvantages of this packed bed bioreactor.
Last, flocculated cell bioreactor is a similar like to fluidized bed bioreactor. It could be a type of a reactor used to promote a flocculation in a water treatment processes. It may involve creating an environment where flocculation can occur efficiently. So basically, flocculation is a process where established particles in a water form larger aggregates called a flocks through the addition of a chemicals known as a flocculants. In advantages and disadvantages, similar like to a fluidized bed bioreactor.
So here we completed our video. I hope you enjoying very well. And if you want to get more videos with this kind of a simplest understanding, join with us, subscribe to our channel, and press the bell icon for never miss an update. Thank you so much, friends.